Hello. Welcome back to Learning Lab Diagnosis under 5 minutes. This video is in continuation with learning the respiratory infections directly from our book Apurva Shastri, and this is the second video about H. influenzae bacteria. This is one of the causes for typical lobar pneumonia. Now let us start. So since this is a bacteria, I need you to focus and imagine what do we do with the bacteria. First of all, we'll take the bacterial sample. All right, for that we need the specimen. So we are doing specimen collection. After that, we are doing direct examination of the bacteria. Then since this is a bacteria, you'll do the culture immediately. After that, identification and antimicrobial susceptibility testing. This is the basic skeleton of any bacterial lab diagnosis that you will come across. There are two additional things here, which are the typing methods and the molecular methods, which I've ticked for you so that you can have a visual memory of it during the exam. So now that we know the black bold headings, the main challenge, how will you remember the inner contents and not get confused in the exam? So now, again, as our usual format, let us take the main word and weave our story around it. So as you can see with the H. influenzae, it very much goes along with the word influencer. So let's remember our H. influenzae is an influencer, and now the entire story will weave around it. So since this is an influencer, it needs immediate processing and cannot tolerate any extreme conditions, so should never be refrigerated and should be processed immediately. Now again, being an influencer, this is a bit on the negative side, okay, not a very good influencer. So gram negative, and it has fear of missing out now, so it has the characteristics of both coccus and bacilli. So coco bacilli. All right, then being an influencer, it has a capsule around it, so capsule, and then it also has antigen. So up till now what we have learned, for the specimens we are taking, since this is a respiratory, we are taking the sputum. Additionally, we are taking blood and CSF, which should be processed immediately, never be refrigerated. Direct examination, three things: pleomorphic gram-negative coccobacilli, capsule detection. One name that you got to remember is Quilling reaction. In antigen detection, by the basic three things that you've always done: latex agglutination, direct IF, or ELISA. Now the culture. So one important thing you remember, this is the influenza. The influencer is coming in a satellite, and back at home the influencer had chocolate fields, which was named as, and this field's name was Leventhals. All right, so this is a culture. So in culture, what are you doing? Blood agar with Staph aureus streak line shows satellitism. Very important to write in the answer of this bacteria. All right. Others in that we have the chocolate field. You have to remember the chocolate agar, fieldies agar, and Leventhals agar. How will you identify? Being an influencer, this has some special factors. Right. So remember the factors, and automated systems common for every answer. Maltitov Vitek. Then for the typing methods, you have to biotyping and serotyping. Molecular method, finally our influencer has made it to the cinemas and we are watching him on multiplex PCR. And in the end, we do antimicrobial susceptibility testing. So quick revision, running you over the story once, and then in the second part of the revision, we'll discuss the points so that I want you to recollect along with me. Going over the story, this is an influencer, so needs to immediately be processed, should never be refrigerated, is a gram-negative coccobacilli, has a capsule and antigen, shows satellitism, chocolate fieldies and Leventhals agar, has some special factors, automated systems, goes into biotyping, serotyping, finally makes it to the multiplex PCR. Okay, now what will you write your answer? Revise along with me. In the specimens, three types of specimen: sputum, blood, CSF, should be processed immediately, never be refrigerated. Second heading, direct examination. Under direct examination, what will you see? Three things: pleomorphic gram-negative coccobacilli, capsule detection by what? Quilling reaction, antigen detection by the basic three: latex agglutination, direct IF, ELISA. Culture, how will you see the culture? It will show satellitism. Others, what will it show? Chocolate agar, fieldies agar, and Leventhals agar is what you will use. Other cultures, all right. Identification, biochemical tests such as, which will show special factors. Automated systems common for everything. Maltitov Vitek. Typing methods such as biotyping, serotyping, molecular method, multiplex PCR, antimicrobial susceptibility testing. That's all. We have done this. Just remember the word influencer and remember the story around it, and I hope you'll not get confused during the exam. Stay tuned for more videos. And also, please learn this as a basic skeleton and study the inner theory parts to expand your answer. This is just a skeleton to aid you to learn the main points, but you have to read in detail to secure the full marks.